name is Julia Vu, and I'm an undergraduate researcher in the Gozani Lab, mentored by Dr. Shou Liu. And my work revolves around understanding the biochemical and genetic basis of, of gastric carcinoma tumorigenesis. Protein synthesis is a fundamental biological process that is known to be heavily regulated and that is essential for cancer cell proliferation. Methyltransferases frequently target protein components of the elongation translation machinery, epigenetically modulating their activity and influencing the fidelity and efficiency of protein synthesis. In particular, GTPase eEF1A, or eukaryotic elongation factor 1 alpha, is methylated at several lysine residues, which contributes to the regulation of translation elongation, and other specific eEF1A functions. Previous work has identified the methyl-13 methylase eEF1A at lysine 55 and stimulates protein synthesis, driving Ras-driven tumorigenesis in lung adenocarcinoma and pancreatic ductal adenocarcinoma. My research shows that methyl-13 dimethylation of eEF1A promotes gastric carcinoma proliferation through increasing global protein synthesis. Methyl-13 is broadly upregulated in many types of cancers, most notably gastric carcinoma, where meta-analysis of public gastric carcinoma datasets indicate that approximately 77% of patient samples contain some genetic alterations to methyl-13 expression, most notably gene amplification and high levels of mRNA. Additionally, according to data from the Global Cancer Observatory in 2022, gastric cancer is the fifth most common cancer and the third leading cause of cancer-related global mortality globally. As a result of late-stage diagnosis in many cases and the limited therapeutic options once the disease has metastasized. Therefore, there is a critical need to identify clinically actionable pathways that regulate gastric cancer tumorigenesis. I conducted a series of cell proliferation assays using two mammalian gastric carcinoma cell lines, N87 and HGC27, to assess the role of methyl-13 in gastric cancer tumorigenesis. Using CRISPR-Cas9, I generated two independent methyl-13 knockdown cell lines and assessed the relative proliferation. I found that methyl-13 depletion results in significantly decreased cell proliferation in both cell lines and independent knockdowns. The Western blots beside the graphs indicate the reduced levels of methyl-13 protein levels, which is paralleled by a decrease in the eEF1A lysine 55 dimethylation signal in both methyl-13 knockdown cell lines. In order to better understand the role of methyl-13 in inhibiting gastric carcinoma proliferation, it's important to understand the significance of lysine residue 55. eEF1A is the small GTPase that delivers amino-acylated tRNA to the ribosomal A site during protein synthesis. The protein itself consists of three domains, most notably domain 1, which is the catalytic GTPase domain. Since lysine 55 is located in GTPase domain, we can make an educated guess that the dimethylation of eEF1A at lysine 55 likely regulates GTPase activity of eEF1A and, by extension, literally, the mRNA translation in the cells. In order to test this hypothesis, I wanted to look at the impact of methyl-13 depletion on global protein synthesis. I conducted an AHA labeling experiment where I grew N87 cells in methionine-depleted and L-acetohomoalanine, or AHA, enriched medium, such that the AHA, which is a methionine analog, incorporates into the newly synthesized protein in place of methionine. I repeated this experiment in cells under normal, serum starvation, and serum stimulation conditions to synchronize the cells to the G1 phase of the cell cycle. Western blot analysis of these cells, probing for AHA, would indicate that in every condition, the methyl-13 knockdown cells have significantly less global protein synthesis. Similarly, I conducted a sunset assay with the HGC27 cells, utilizing pyrimycin, which incorporates into the growing polypeptide chain by resembling an amino acid attached to the tRNA. These results, although a little bit more subtle, also corroborate that methyl-13 depletion decreases global translational output. Overall, my work demonstrates the methyl-13 dimethylation of eEF1A at lysine 55 plays a fundamental role in promoting gastric carcinoma tumorigenesis and protein synthesis. The upregulation of methyl-13 in cancer patients also suggests that there may be a therapeutic window to target methyl-13 in gastric carcinoma and a broad range of other human cancer types. Moving forward, I hope to conduct a clonogenic assay, to visually assess if methyl-13 impacts colony formation in gastric carcinoma, as well as attempt to replicate these results in 3D cell culture, which would more accurately reflect the tumor microenvironment. Once again, I would like to thank Orr and the entirety of the Gozani-Chua lab, but most notably Sho, for taking me in as a little high schooler who barely knew how to hold a pipette, and for being one of the biggest reasons I want to spend the rest of my life pursuing research. Thank you.